Hello everyone and welcome to today's video. In today's video, we're going to be talking about the Pfizer, BioNTech / Moderna mRNA vaccine technology. Essentially, in this video, we're going to be talking purely on the mechanism of action of this vaccine, and we're not going to be talking about its efficiency or any of that stuff, and it's going to be covered within future videos. So let's get started. Essentially, this vaccine is going to be present, the doses are going to be present in vials, and these vials are going to be containing lipid nanoparticles, and these lipid nanoparticles are going to be having mRNAs that are going to be present within them. Those mRNAs are going to be coding for the S protein or spike protein that is characteristically present on the surface of the SARS-CoV-2. Essentially, this vaccine is going to be injected using a syringe into the arm of people that want to get vaccinated. Now, upon injection, these lipid nanoparticles are going to be migrating and they're going to be fusing with the cell membranes of the host cells, as we see here. Essentially, upon fusion of the lipid nanoparticle to the host cell, it is going to be releasing the mRNA into the cytoplasm of the host cell. Now, this is going to be leading to the mRNA to use the translational machinery and the ribosomes present within the cytoplasm to produce the protein, the S protein that it is going to be coding for. Now, it's very important to note, and I'm not going to go into much details, we'll leave that to a future video. However, the mRNA in this scenario is not, and I repeat, it does not migrate to the nucleus and it does not incorporate into our genome. That being said, so we're going to now be having the S protein or the spike protein within our cytoplasm. This is going to be leading the MHC class 2 and the MHC class 1 to express this protein onto its surface as antigens. Now, this is what we are going to be seeing in this image right here, or in this part of the mechanism, where this protein, the spike protein present and represented by this small ball, it's going to be present on the MHC class 2 and also the MHC class 1, but I'll leave that till the end of the video. So this is going to be recognized by the TCR-CD3 cells, the T helper cells, also called the CD4 cells, and this is going to be leading the T helper cells to release cytokines. These cytokines are going to be having two major roles. First role is going to be to stimulate the proliferation of the B cells that are going to be leading to the formation of plasma cells that are going to be releasing antibodies that will neutralize the virus when it detects it. And also the cytokines are going to be stimulating the proliferation of memory T cells, and this is also going to be helping in the immune response against the virus. So let's repeat that. We're going to be having the cytokines stimulating B cells, giving us plasma cells, releasing antibodies. Also, we're going to be having memory B cells for the future recognition of this virus. Also, we're going to be having T cells that are also going to be implicated in the future response to this virus upon exposure. Now, we mentioned that this antibody, this antigen, specifically talking about the S protein, is also going to be expressed on the MHC class 1. Recall that the MHC class 1 is going to be present on all nucleated cells within the body. It's very important to note that the cytotoxic T cells are the ones that are going to be detecting those MHC class 1 receptors having those spike proteins, and they're also implicated in helping the immune response against this virus. This concludes this episode on the mechanism of the mRNA vaccine technology. If you have any questions regarding it, feel free to send them and write them in the description down below. And this has been Ali from BeBiology.